In this video, we're going to talk about DNA replication, and this is going to be IV sections uh, 3.6 and I think 7.6. Looks like that got cut off. Now, within DNA replication, the first thing that we want to look at is how DNA is replicated. Specifically, meaning how are these strands that are created? How do we get two strands from one strand? And this is something that we call semi-conservative. The DNA replication process is semi-conservative. And what that means is that we've got, there originally were a couple different ways that we thought DNA was replicated. Um, and if we look at these here, we've got semi-conservative, conservative, and dispersive. In dispersive, our original strand here is in the red, and so here's our dispersive strand. And under this idea, the original strand is basically mixed up with our newly synthesized strand. So this is dispersive. We've got a mix of both. In our conservative strand, here's our conservative strand. We've got our original strand, and from this we have two strands, and one of them is completely the original strand, and then the other one is our newly synthesized DNA. The third option, and the one that we've found to actually be most accurate in our studies of DNA and how it's replicated, is called semi-conservative. And this is what you need to know, is that DNA replication is semi-conservative. And in semi-conservative, we have our original strand, and for each original strand we get a template strand. So we've got both here. We've got our original strand and our new template strand. DNA replication occurs in the 5' to 3' direction. When we've talked about our DNA structure, we've talked about how the sugar, there's 5 carbons on that sugar, and so our DNA structure is actually arranged in a 5' to 3' direction. Well, DNA has to be added in a specific 5' to 3' direction. And so as we start to look at some images here, we'll see specifically what this means and how that works. Here's an image that can kind of help to illustrate that. Here's our original DNA strand. Here's our original DNA strand here as well. And in this image is showing us how DNA is replicated. Well, we're moving towards the 5' end. So this portion of the DNA template strand here is the 5' end. This portion is the 3' end. And so they're kind of going in opposite directions, you could think of. This one's moving in the 3' direction. This one's moving in the 5' direction. Replication has to occur, or new nucleotides have to be added in the 5' direction. So we can see right here, and we'll take a look at this more specifically in a minute, nucleotides are being added in the 5' direction. On this other strand, nucleotides are being added in the 5' direction, moving this way right here. DNA replication is initiated at many different points in eukaryotic chromosomes. There's not just a single point where replication is started. There's actually multiple points within a chromosome. And what we see happen is those are initiated at multiple points, and once they kind of meet each other, they fuse. And so we kind of have like two little bubbles that come together and become one. And then the last portion of the DNA is replicated, and that chromosome is complete. Replication begins at the origin of replication. That's a specific spot. It's a specific section of DNA, or a specific gene section that initiates replication at that portion. There's some interesting research being done of discovering exactly how this works. For our purpose, you just need to know that replication begins at the origin of replication. Within DNA replication, there's something called complementary base pairing. And base pairs always match up in the same fashion. We always have our adenine and thymine matching up, and our guanine and cytosine matching up. So those nitrogen base pairs are always pairing up together, adenine and thymine, guanine and cytosine. An incorrect matching would result in a mutation. So adenine and thymine are most generally always going to line up together and to be added together. If there were to be a switch, or something were to occur that would force adenine and maybe cytosine to match up, that would be a change to the DNA sequence. That would be a mutation. Now, as we've talked about with introns and exons, that may not necessarily cause a huge overall change to the protein that's produced, or to whatever the gene is responsible for. If it occurs in an intron, it's probably not going to have a huge overall effect, but it still would be considered a mutation. Complementary base pairing allows DNA molecules to be identical. And this is what's really important here is that through DNA replication, we're making two strands from one that are identical. Just like how we talked about with the cell cycle, and at the end of the cell cycle, we have two cells that are identical. They have the same DNA because that DNA is replicated during S phase of interphase before the cell actually divides, and so we get two strands from one that are identical, and those two strands are semi-conservative. One original parent strand of DNA and one template strand. And then lastly, new strands are complementary to the templates. So we have a new strand that matches with the original or the parent strand that's complementary to it. And it's also identical to the other template. Let's say we have our two strands of DNA. They split apart to become replicated, and so the templates are going to match the opposite original strand. So let's kind of talk through the process of DNA replication. As we talked about it, it occurs in the 5 to 3' direction. And as we mentioned before, there is kind of a flip-flop of how DNA is set up. If you think back to when we used, or actually we will be using little puzzle pieces of nucleotides to sequence the steps of replication. And when you're doing that, you'll have to actually turn upside down one strand. That would be like the 3' direction strand in order to have the nucleotides, the bases, fit together. One of the first things that happens during DNA replication is helicase here. This is an enzyme. And helicase is actually what is responsible for breaking apart our DNA molecule. It's splitting those two strands in half. It's breaking the hydrogen bonds that are holding these nitrogen bases together. We've talked about nitrogen bases, and the helicase is what's separating those two. Once the DNA has been separated, we have two different strands here. We have the leading strand and the lagging strand. And let's talk about the leading strand first. DNA polymerase III is the molecule that's responsible for adding new nucleotides to our original strand. So in our leading strand here, the green is our original, or the parent strand you could think of, and the yellow are the new nucleotides that are being added. Polymerase comes along here and adds new nucleotides to our strand. And in order for this to actually occur, we have to add a little bit of RNA primer. RNA primer is added so that it indicates DNA polymerase to actually add nucleotides. It's kind of the starting mechanism, and that occurs in the lagging strand here that we'll talk about in just a minute. So this is our leading strand. We can see that this is in the direction of the 5'. So we're adding new nucleotides in the 5' direction. It's our leading strand. The lagging strand's a little bit more complicated. What we're seeing in our lagging strand is because we're moving in the 5' direction, nucleotides have to be added a couple at a time and then put together. And those couple at a time are something that we call an Okazaki fragment. And those are named after the scientists who discovered this. And so basically what we're seeing happening is nucleotides are added, a series of nucleotides are added by DNA polymerase after an RNA primer has been laid down. They're added, and then what we're going to see happen is DNA ligase kind of goes backwards and kind of puts all of those Okazaki fragments together. And so in our lagging strand, these are put together in kind of a fragmented section. We have little short sections called Okazaki fragments that are added, those being nucleotide bases, and DNA ligase comes along and fixes those or puts those together so that we have a full strand. Let's take a look at the steps here, a little bit more detail. I'll come back to DNA polymerase in just a second. Steps to DNA replication. So the first one is that helicase uncoils the DNA. RNA primase, adds short sequences of RNA to both strands. This is called the primer. This is kind of like what's initiating it. If you think about if you were to paint something or if you were to paint a house, oftentimes you'll use a primer as kind of a base layer. That's to help kind of smooth over all the painting process and make it a higher quality when it's done. RNA primase is kind of the same thing. It's laying the groundwork. It's initiating the process of adding DNA nucleotides. The primer allows DNA polymerase III to bind and start replication. DNA polymerase III adds nucleotides to each template strand, or our parent strand, our original strand, in the 5' to 3' direction. And nucleotides lose 2 phosphate groups to release energy. So this process does require some energy, and these nucleotides, when they're being added, they lose 2 phosphate groups to provide that energy. One DNA strand, as we discussed, is replicated in a continuous manner, and this is in the same direction as the replication fork. So if the replication fork is moving towards that 5' direction, nucleotides are going to be added in that same direction, and we call this the leading strand. The second DNA strand is replicated in fragments, and it's moving in the opposite direction. We call this the lagging strand, and this is where we see the Okazaki fragments being added. So if we're moving in the opposite direction, this is the lagging strand. We're still adding nucleotides in that 5' to 3' direction, but because we're going backwards or opposite, they have to be put back together. Those fragments have to be put back together, and those are called Okazaki fragments. DNA polymerase I removes the RNA primers and replaces them with DNA. And then DNA ligase is joining those Okazaki fragments in the lagging strand together to form one continuous strand. So that's only occurring in the lagging strand. The leading strand moves at a continuous manner in the same direction. Let's go back quickly and take a look at our DNA polymerase. DNA polymerase is a really interesting molecule as it's adding new nucleotides. So as we're adding new nucleotides here, we're extending our new strand here. We're adding new nucleotides, and so if we've got an A and a C here, an adenine and a cytosine. The A and the C, they don't match up. That would obviously be a mutation if this were to stick. DNA polymerase has this really cool ability to kind of do some proofreading, or kind of like if you were word processing, kind of like a spell check. As it's going along, it proofreads the addition of new nucleotides to our original template strand. And so in our image here, DNA polymerase catches this as an error. A and C don't match together. This should be a guanine that matches here. It removes that nucleotide, and then when the guanine is available, adds it to match with the cytosine. So DNA polymerase has some abilities to actually do some proofreading and do some checking. And the majority of errors that do occur during DNA replication are usually fixed by DNA polymerase as it's adding nucleotides. That's a quick overview of DNA replication. The important things to remember is that there's 2 different ways that DNA is added, the leading strand and the lagging strand, and being familiar and able to understand and explain how Okazaki fragments are created and then put together using DNA ligase. So that's DNA replication. We'll get a lot more practice in class, kind of walking through the steps with some models and some diagrams.